The possibility of generating different subtypes of motor neurons is particularly relevant for modeling motor neuron diseases such as amyotrophic lateral sclerosis. This method allows the acquisition of cell populations that are highly enriched for motor neurons with a spinal or a cranial identity in a short time frame and without a purification step.
The generation of human motor neurons from induced pluripotent stem cells, or iPSC, under simplified culture condition may facilitate drug screening for motor neuron diseases. The inducible expression of programming transcription factor can be used to generate another cell types, such as neuronal skeletal muscle and glia cells, from an iPSC repository.
For nil and NIP iPSC line generation, rinse the human iPSC culture with calcium and magnesium-free PBS before treating the cells with cell dissociation reagent for 5 to 10 minutes at 37 degrees Celsius. When the cells have begun to detach from the culture container, use a P1000 pipette to gently manually dissociate the cells 3 to 4 times. Transfer the cells to a 15 milliliter tube and bring the final volume up to 10 milliliters with fresh PBS without calcium and magnesium for counting. Collect 1 × 10⁶ cells by centrifugation and resuspend the pellet in 100 microliters of buffer R from the electroporation kit. Add 4.5 micrograms of transposable vector plasmid DNA and 0.5 micrograms of the PiggyBac transposase plasmid DNA for transfection.